Hello everyone. I would like to thank the organizers for this great opportunity to present my work here today. I would like to present here some preliminary data regarding the use of protein language models to predict the functions of proteins and how this technique has been demonstrated to do surprisingly well within the recent Kappa-5 protein function prediction competition. Proteins, as we all know, are these fundamental molecules that play a crucial role in underpinning the complexity of life. They come in an incredible diversity of forms that carry out all the functions necessary for life. So what are proteins exactly? Proteins are formed by linear chains of amino acids, which in turn form complex secondary, tertiary, and quaternary structures through three-dimensional folding. Given the importance of proteins in biological processes and their complexity, predicting the functions of novel and understudied proteins remains an open challenge within the field of biology. One way to represent protein functions is through gene ontologies, where we assign specific terms to a protein based on knowledge that we have about its function. There are three gene ontology domains: molecular function, cellular component, and biological process. We can see here that more specific goal terms, such as hexose biosynthetic process, implies the existence of all parental terms from that goal term for that protein. Traditionally, protein function prediction has been quite challenging. Existing methods rely primarily upon sequence alignment and motif analysis based on the principle that proteins with similar primary sequences will carry out similar functions. Other information, such as protein-protein interaction, have also been demonstrated to be helpful in predicting function. This is a figure from the NetGo 2.0 paper from the Zhu lab that shows how an ensemble strategy of various protein information is able to produce very good prediction of protein function. One challenge in the field of protein function prediction is the problem of incomplete data. Notably, of all the proteins with experimentally verified predictions in UniProt, almost half of them only have annotations from a single ontological domain, when we can realistically expect almost all proteins to have some conserved ontological role in all three domains. Now, in order to find the best new method for predicting protein function, the Kappa-5 challenge, which is ongoing, seeks to evaluate protein prediction algorithms against a vast number of test proteins, 141,865 to be exact. Of those proteins in the test set, a small subset of them will be used for the final evaluation by looking at newly discovered, experimentally verified functions of those proteins after the final submission is over. In order to tackle this problem of protein prediction, we use as our baseline method protein language models for predicting function. These large protein models are based on the transformer architecture and encode amino acid sequences into distributed vector representations that capture the structural and functional properties of proteins. This allows these language models to be tuned to predict a variety of different protein characteristics, including protein function and gene ontology. Currently, the state-of-the-art protein language models such as PROT5 and ESM2 utilize a mask language modeling objective, which essentially masks certain amino acids during training of the model, and then the model is trained to predict the identity of the masked amino acid. These pre-trained models are used to produce sequence-level embeddings that encode essential information about a protein sequence, which is what we use during training and testing in this competition. Our approach thus utilizes these protein language models to produce a set of predictions of goal terms that perform surprisingly well in a Kappa-5 competition. We observed that biological process goal terms are harder to predict compared to cellular component and molecular function based on the Fmax score. Overall, out of over 1,000 teams who have made predictions in this competition, our current rank on the leaderboard is third, which indicates that our model performs fairly robustly compared to its peers. We can imagine that given the ability of these language models to predict the functions of proteins, it may now be increasingly possible to utilize such models to invert this pipeline and predict protein sequences based on a predefined set of functions that we wish a protein to carry out. Indeed, there are currently many research groups that are pursuing such approaches, and it's very exciting to see the emergence of this new technology and its potential applications in deepening our understanding of protein design and function. In conclusion, I would like to thank the lab, my PI, Peter Adams, and in particular Adarsh Rajesh for helping out in this project, as well as Sanju Sinha for guidance. Thank you.